structure of Z form of DNA. So before moving into the detail of Z form of DNA, we must know what is the double helical structure of DNA. DNA is a double helix and it's basically look like a twisted ladder. As you can see in the image, the sugar phosphate backbone. This is a sugar fast phosphate backbone, and the base pairs form the interior. The two phosphate backbones are basically anti-parallel to each other, which means that if one backbone runs from 5 prime to 3 prime direction, the other runs from 3 prime to 5 prime direction. There is also a major groove and a minor groove. Major groove is where the phosphate backbone are far apart, and minor groove is where they are close together. Now, when Watson and Crick gave the model for the structure of DNA, they said that the distance between two base pair is about 3.4 angstrom, and there are about 10 base pair per helix. But later it was discovered that there are about 10.5 base pairs per helix, giving a helical length of 36 angstrom. Now, moving on to the different forms of DNA, and we will see that how zDNA is different from aDNA and bDNA. As you can see in the image, that aDNA and bDNA are both right-handed, means the backbone, the phosphate backbone, runs from right to left, whereas in zDNA, the backbone runs from left to right. The zDNA diameter is narrow or short, but it is more, the zDNA is more stretched out, means its helical length is much more, as you can clearly see. The minor groove in zDNA is narrow and deep-seated, whereas the major groove is nearly absent. zDNA was discovered by Alexander Rich and Andres Wong, and it is generally found at the start of the gene, and hence it is believed that it plays some role in gene regulation. We'll see further how it helps in gene regulation. So now let us see the conformation of zDNA and how it is formed from bDNA. See what happens in bDNA, the two base pairs are both in anti-conformation, as you can see here. So they form a mononucleotide nucleotide. While what happens in zDNA, the pyrimidine that or the pyrimidine unit uh, is in syn or anti-conformation, uh, and the purine is in syn conformation, as you can see here in the image. So this zDNA forms a dinucleotide. Now in bDNA, both G and C are in C2 dash endo conformation, whereas in zDNA, C is in C2 dash endo conformation, and G is in C3 dash endo conformation. Now, what is this C2 dash and endo and C3 dash endo? As we know that DNA has a sugar, has a sugar molecule and has a sugar structure. So not all the molecules are in plane. So in C2 dash endo, the second carbon is raised from the plane, whereas in C3 dash endo, the third carbon is raised from the plane. Now, this anti and syn conformation gives zDNA its zigzag look, and hence its name came from here, zigzag, the zDNA. Now, coming on to the stability of zDNA. zDNA, as we all know, due to its structure, is relatively unstable. So it is generally stable in the presence of high salt concentration like calcium and magnesium. Now you must be thinking why high salt concentration is required. High salt concentration is required because, as we have seen, zDNA has only minor grooves. Minor grooves means the phosphate groups are very close to each other. So there would be a lot of negative charge. So to counter that negative charge, some positive ions are required to stabilize zDNA. And it is also stable in presence of molecules such as spermine and spermidine. Now, it was also seen that negative supercoiling stabilizes zDNA. Now, why so? Uh, See what happens when bDNA is transformed to zDNA, there would be a lot of energy required, and hence zDNA is unstable. Now, when negative supercoiling requires energy and it tends to unwind bDNA. Now, when this process releases energy and hence stabilizes DNA. Now you can see in this image, in the first image, there are about three negative supercoil, but after two negative supercoil decreases, and hence one Z DNA segment is formed. Okay, now coming on to the biological activities of zDNA and what's its role in biology. Now, as we have said, that zDNA is found at the start of gene, where the promoter region is present. And we all know that the promoter region is the start of basically the transcription. So it is hypothesized that zDNA has some role in what you say, this transcription. So this is example of MYC gene. MYC gene is basically a regulator type of gene which regulates transcription. So what happens when RNA polymerase comes to the promoter and starts the transcription, it leads to the unwinding of DNA. Unwinding of DNA means negative supercoiling. And as we have seen that negative supercoiling stabilizes zDNA. So zDNA is formed at the start of the gene, and hence it is believed to have some role in transcription. A study of human chromosome 22 also showed a correlation between zDNA forming regions and the promoter regions for nuclear factor 1. This suggests that transcription in some human genes may be regulated by zDNA formation and nuclear factor 1 activation. Now, you must all have heard about nucleosomes, and nucleosomes are present at the start where the transcription starts. But zDNA do not form nucleosome because of its complex zigzag structure. So it is believed that zDNA is hypothesized to code for nucleosome positioning, and hence it regulate the rate of transcription. Moving on, the interaction of different kinds of protein with zDNA. E3L is one kind of protein that interacts with zDNA. E3L protein is basically a protein found in is a vaccina virus protein, for pox virus protein basically. As zDNA has been researched more thoroughly, it has been discovered that the structure of zDNA can bind to zDNA binding proteins through London dispersion and hydrogen bonding. One example of a zDNA binding protein is the vaccina E3L protein, which is a product of E3L gene and mimics a mammalian protein that binds zDNA. Not only does the E3L protein have affinity to zDNA, it has also been found to play a role in the level of severity of virulence in mice caused by vaccina virus, a type of pox virus. Two critical components to the ethereal protein that determine virulence are the N terminus and C terminus. As you all know that protein is basically amino acid. So amino acid has a N terminus and a C terminus. So this is talking about that. The N terminus is made of up a sequence similar to that of the Z alpha domain, also called as adenosine deaminase Z alpha domain, ADAR. While the C terminus is composed of double-stranded RNA binding motif. Through research done at various laboratories, it was shown that replacing the N terminus of the E3L protein with a Z alpha domain sequence containing 14 Z DNA binding residues similar to E3L had little to no effect on the pathogenicity of the virus in mice. In contrast, it was also found that deleting all 83 residues of the E3L N terminus resulted in the decreased virulence. This supports a claim that the N terminus containing the zDNA binding residues is necessary for virulence. Overall, these findings show that similar zDNA binding residues within the N terminus of the E3L protein and the Z alpha domain are the most important structural factors determining virulence caused by the vaccina virus. While amino acid residues not only involved in zDNA binding have little to no effect. A future implication of these findings includes reducing zDNA binding of E3L in vaccine containing the vaccina virus so negative reaction to the virus can be minimized in humans. Overall, these results show that the decreasing the bonds and interaction between zDNA and zDNA binding proteins decrease both virulence as well as genes expression. Hence, this was the importance of zDNA binding with E3L protein. So in conclusion, I would like to say that as we have seen that zDNA has very complex structure where there is anti-conformation as well as syn conformation, and it is not very stable. Basically, it exists in meta stable state. There have been researches going on to know much more about zDNA, but what we have seen has provided insights that are likely to stimulate more research into this unusual left-handed version of the DNA double helix. Thank you.